Okay, I think we might get started. Casey, is that coming through okay, just to make sure?
Yes, you're good, Doug.
Beautiful, thank you, and good morning and a very warm welcome to everybody, wherever you are, and an especially warm welcome if you are joining us from Sydney or Brisbane, and our thoughts are with all of you further north as it continues to rain. It's my absolute pleasure to be with you today to formally launch a really important facility, the ACRF program for resolving cancer complexity and therapeutic resistance. I'd like to begin by acknowledging and celebrating the traditional owners and custodians of the land on which I am joining you from today, here in Parkville in Melbourne. And they're the Wurundjeri people of the Kulin Nation. And I pay my respects to their elders past, present, and embrace their continued connection to this place. I'd also like to extend this respect to elders from other communities where people are joining us from today. And I'd also like to celebrate the wonderful contribution Aboriginal and Torres Strait Islander staff and students make to creating a vibrant campus and are making a wonderful contribution to cancer research here at WEHI. I'd like to extend a special welcome to the Australian Cancer Research Foundation supporters. Thank you for being with us today and for your really long-term passionate support of medical research into cancer. And I just want you to know you are making a real difference in the work that our scientists can do, and through them, the outcomes for patients with cancer in the community. Welcome also to a friend of WEHI, Mr. Russell Kaplan, who has been on the board of trustees at ACRF for over 10 years. And additionally, I'd like to welcome other members of the ACRF board and also their dedicated staff, notably Kerry Stradham, Ian Brown, Carly Dutoit, and Cheryl Riddington, and say a very big thank you to all of you for your support of cancer research, not just at WEHI, but across the Australian biomedical research sector. Further to this, we're very fortunate to have also received a valuable grant for this project to support the salary of a postdoctoral bioinformatician, that is a mathematician who helps work on biology and creates different ways of handling data, and allows, through their creativity, us to derive really good insights about how cancer arises. That bioinformatician is necessary to help us operate the sophisticated software required for the new analyses of both single-cell genomic and imaging data, which is produced by the equipment that was the centre of this new grant. Also, a very special thank you to Lucinda Nolan and the scientific committee at the Ovarian Cancer Research Foundation, who collaborated with Australian Cancer Research Foundation to better resource our project, because equipment is important and crucial, but having the technologists who run that equipment is also a really key need if we're going to be successful. Finally, a welcome to my team, that is our WEHI scientists who've made important contributions to cancer research over the years, and in particular to the investigators and their team that made this program possible. Just a couple of housekeeping items. Shortly, we will be conducting a panel discussion with some of the scientists involved in this new ACRF program. And if you would like to ask the scientists any questions, you can do so at any time by submitting your questions in the Q&A box at the bottom of your Zoom screen. WEHI's had a long and successful history of cancer research. And it began way back in 1923 when Dr. Marion Boyd Wanless joined WEHI. Dr. Wanless was studying cancer of the uterus, which is a malignant disease and affects certain birds. And these tumours hold similarities to the tumours found in women. At the time, WEHI director, Charles Kellaway, said that Dr. Wanless's work was probably the only cancer research being currently undertaken in Australia. And it's exciting to think that WEHI's journey stretches back those 99 years to Dr. Wanless's work on uterine cancer. Fast forward nearly 100 years and our cancer research continues to flourish. We currently have over 450 researchers focusing on the most significant cancers in Australia, such as blood, brain, breast, lung, ovarian, bowel, pancreatic, and rare cancers. What makes this exciting is that these exceptionally bright minds are committed to solving the most complex problems in cancer and making transformative discoveries, not alone, but in wholehearted collaboration. And we hope that these discoveries help people live healthier and for longer. We thank the Australian Cancer Research Foundation for their generous support and indeed your long-standing faith in our ability to understand, prevent and find new treatments for people with cancer. Over the years, ACRF grants have enabled WEHI to invest in people, infrastructure and technology that advance our cancer research. We've been able to establish a significant number of research initiatives with our Chemical Biology Division, Cancer Biology and Stem Cell Division, the ACRF Breakthrough Technology Laboratory, and now we'll launch the ACRF program for resolving cancer complexity and therapeutic resistance. This program will specifically address one of the main conundrums of precision medicine, and that is how to deal with the inherent heterogeneity or variability of cancers. That is, how can we approach the differences between tumours of the same type in different patients, and indeed by cells of different types in the one tumour of an individual patient? And how can we in turn determine the variability of how patients respond to therapies? We know as a tumour progresses, DNA replication becomes unstable and mutations develop. Our challenge is to address this and the related problem of cancer treatment resistance, including emergent resistance to targeted small molecule and biological therapies. With the assistance of this generous grant from ACRF, WEHI has been able to purchase state-of-the-art equipment that will allow us to investigate the cancer environment at a single cell level and provide unprecedented insights into the evolution of these cancer-driving cellular changes with disease progression and response to therapy. We can also identify common pathways as well as processes to understand specific alterations in an individual patient's cancers, allowing further opportunity to refine treatment and individualized care. Importantly, this grant also brings together 21 investigators and their teams from across WEHI to contribute to this project. These scientists have expertise across a really broad range of disciplines. And just to name a few: biology, drug discovery, technology, genetics, clinical care, bioinformatics, and importantly, treatment resistance. WEHI and its researchers are well known for their collaborative working style and culture, both throughout the Institute, across the Parkville precinct, across Australia and around the world. A focus on collaboration, on bringing together big multidisciplinary teams to work often for years and decades on solving significant health challenges is the corner of WEHI's research strategy. Our ability to do this, to have the vision for the future that is courageous, collaborative, ambitious, is in part thanks to the support we receive from ACRF. It's thanks to your grants, recognitions, award and support, and your belief in the transformative impact of our research that our scientists can work in an environment that enables them to flourish, encourages them to be bold, and nurtures their curiosity. You've contributed to the building blocks of scientific knowledge, which ultimately is what leads to improvement in health outcomes, helping people with cancer live healthier lives for longer. Now is when I would usually invite you on a tour of our laboratories to see the equipment that we've been able to purchase with your generous support. Unfortunately, until very recently, COVID-safe restrictions have put a halt on our ability to have in-person visits. So we have an alternative way to show you the equipment purchased with the ACRF grant that will progress our understanding of cancer complexity and therapeutic resistance. And I hope you enjoy this short video that we put together. Hello, I'm Professor Doug Hilton, Director of the Walter and Eliza Hall Institute. ACRF has been a supporter of WEHI for over 20 years. We've had 6 successful grants that have allowed WEHI to purchase important equipment that advances our cancer research. With ACRF's most recent support, WEHI has been able to launch the ACRF program for resolving cancer complexity and therapeutic resistance. This program will bring together cancer biologists, clinicians, bioinformaticians, computational biologists, and technology experts. It will provide them with a huge range of new research tools to enable them to resolve cancer complexity and therapeutic response in individual patients at the single cell level. The program will specifically address one of the main conundrums of precision medicine. How do we deal with the diversity of cancers and the variability of a patient's response to medicines? We'll also address the related problem of cancer treatment resistance. The ACRF program for resolving cancer complexity and therapeutic resistance has enabled WEHI to purchase cutting-edge technological infrastructure that are essential for the cancer research goals of our ACRF program. The equipment includes the MIBI-scope, the fastest, highest resolution mass spectrometry-based imager ever made. It's the only machine that is capable of the analysis scale, sensitivity, and large sample size proposed in this program. This platform will greatly enhance our opportunities for discovery of unique patient cancers, enabling a greater understanding of the complexity and opportunity for treatment. WEHI has also been able to purchase a multi-photon microscope. This advanced microscope will allow researchers to look inside tissue to study cancers growing and changing over time. There are 21 WEHI researchers listed on the ACRF program, each with their own research teams dedicated to understanding and improving cancer diagnosis and treatments. These are the brightest minds working on breast, blood, ovarian, bowel, and lung cancers. In their capable hands, with the support of ACRF donors, they will make significant discoveries that will improve the lives of people with cancer. ACRF and WEHI have had a long and successful relationship. ACRF donors have enabled not just this amazing program, but many other equipment needs and support that has ensured WEHI scientists continue to make discoveries for humanity. Thank you to all the ACRF donors and supporters who've made this research possible. Thank you for once again for being here with us today, and I hope you liked the look of those wonderful pieces of equipment. Thank you for supporting our cancer research and our scientists, and I'd now like to invite Mr. Russell Kaplan from the ACRF to say a few words. Russell.
Thank you, Doug. And can I also acknowledge the traditional owners of the land on which the astonishing Parkville precinct sits, the wonderful team from WEHI and the loyal ACRF supporters who are online. It's my pleasure to be with you. And I extend apologies from our ACRF chair, Tom Derry, who's overseas, but otherwise would, of course, have been with us. At the Australian Cancer Research Foundation, we know that 1 in 3 Australians will be diagnosed with cancer in their lifetime, and that the remaining 2 will be affected closely by a diagnosis. And we know also that it takes research and brilliant ideas coming from the best scientists to tackle something as big as cancer. And the scientists tell us that where they can most use targeted help is in the provision of the technology, equipment, and infrastructure that they need for their pioneering research. And that's our mission at ACRF. We provide grants to support technology, equipment and infrastructure vital to progress the cancer research that seeks to progress and transform the way that we prevent, detect and treat all cancers. And by funding research, we're committed to our vision of one day reaching a world without cancer. Since 1984, the ACRF has enabled 78 research projects through 42 organisations, an investment of over $165 million into cancer research across Australia. And that has been made possible by our wonderful donors and supporters who share our determination to facilitate the innovation that will ultimately improve many lives. And I want to take the opportunity here to thank them, one and all, sincerely for their ongoing support. At WEHI and in the wonderful surrounding precinct, we're privileged to have a world-renowned powerhouse at the leading edge of the fight against cancer. And so it's no surprise that this is ACRF's fourth grant to WEHI, now totaling $9 million, starting in 2001 with $1 million to fund X-ray crystallography. In 2010, $2 million to expand the stem cells and cancer division and chemical biology division. In 2014, $2.5 million. I think I said thousand earlier, but I mean million in each case, for the ACRF Breakthrough Technologies Laboratory, and now this $3.5 million grant to fund the ACRF program for resolving cancer complexity and therapeutic resistance. And there have been other grants, and I think Doug mentioned up to 6, including to Peter Mac and to the VCCC, the Victorian Comprehensive Cancer Centre. So Doug, we're proud to support your work. And on behalf of ACRF, I extend our very best wishes to WEHI for your ongoing success.
Thank you so much, Russell. We absolutely appreciate the work that you and the ACRF board members do and the staff. And as you said, most importantly, the generosity of the donors throughout Australia. You really do make it possible for our researchers to make amazing discoveries. And I'd now like to introduce you to 3 of those wonderful researchers who were the lead investigators on this grant. And they're Professor Andrew Roberts, Associate Professor Marie-Liesse Asselin-Labat, and Associate Professor Edwin Hawkins. And after a short introduction, we'll conduct a panel discussion where you can ask your questions of the researchers. And as I said, if you'd like to submit a question, please do so at any time using the Q&A function at the bottom of the Zoom screen. Andrew, over to you.
Thank you very much, Doug. And if I could begin by also thanking all at ACRF for their support with this grant and over the years. So Russell and other members of the board, thank you very much. And to Kerry and her team, thank you very much. And also to the Ovarian Cancer Research Foundation for their support of one aspect of this grant. So my name's Andrew Roberts. I'm a haematologist here in Melbourne, cancer theme leader at the WEHI, and also treat people in Royal Melbourne Hospital in the Peter MacCallum Cancer Centre. My emphasis is blood cancers, and this grant will make a big difference in terms of how we better understand some of the toughest to treat leukemias, lymphomas, and multiple myeloma. With me on the panel is Associate Professor Marie-Liesse Asselin-Labat, and I'll turn over to Marie-Liesse so she can tell you a little bit about herself.
Thanks, Andrew. Yeah, thank you for the opportunity to be here, and really thank you to ACRF and OCRF for their support of our research program at WEHI. So I am joint head of the Personalized Oncology Division, and I lead a research program on lung cancer. In my lab, we are specifically interested in understanding the interactions between tumor cells and the cells that surround those tumor cells. We know that tumor cells evolve over time, and this evolution is influenced by the microenvironment or the niche in which those tumor cells reside. So with the technologies funded by Acrf, we can now map in detail the spatial and molecular interactions between these different cell types within the tumors. And from this work, really, we're hoping to identify pathways that can be activated or blocked to prevent tumor cells from becoming more aggressive or from becoming invisible to the immune system. So thank you very much for your support, and I'll hand over back to Angel. Thank you.
Thank you, Marie-Louise. And also with us, Associate Professor Edwin Hawkins. Edwin, do you want to say a couple of words, introduce yourself?
Hi, everyone. I'm Edwin Hawkins, and I'm a laboratory head in the inflammation division at WEHI. Just want to start by echoing the thanks from Doug, Marie-Louise, and Andrew about the continued support of Acrf. It's a cornerstone of pushing the technology and the progression and understand the biology is the support of foundations such as this letting us buy really expensive equipment. And it's really the intersection of the equipment and the biology together that I think is going to drive fantastic discoveries, and we're going to reach new places we've never done before. My research focus is on immune cells. And when many of those immune cells become mutated, they become blood cancers, hence my massive interest in this field, but also the intersection of how the immune system might detect them as well. And how I study that is using powerful microscopes, which are often very expensive, to conduct quite technical experiments. And in these cases, looking inside living animals and looking at cancer cells and how they interact and grow. That's my research focus, and the Acrf funding helped supply the multi-photon microscope that is the cornerstone of the discoveries made in my laboratory. Andrew, back over to you.
We'll come back to the multi-photon, and I'll ask you in a little bit about how you use it, etc. But just for everyone's knowledge, this is there's only 3 researchers here in front of you, but we actually represent scientists in 21 research laboratories at WEHI and with our external partners. And as you heard earlier, covering some of the toughest cancers within groups, including blood cancers, ovarian cancers, lung cancers, breast cancers, gastrointestinal cancers such as pancreatic cancer and bowel cancer, brain cancer, and various rare tumours. We've all come together in this, what we call the Acrf program for resolving cancer complexity and therapeutic resistance, because there's a central problem with these doing better in treating some of these very different cancers, and that is they're complex. So behind me, you can see a picture of what one type of cancer looks like. Now, it doesn't matter what type of cancer it is, but this is something that we would just call a standard cancer. And for a long time, people have treated cancers as if every cell in a cancer is exactly the same. But as Marie-Louise will explain in an example as we go through some of the discussion, nothing could be further from the truth. In fact, within just about all cancers, there is a mix of cells. Some are normal cells and some are cancerous. Some are immune cells trying to fight off the cancer. Other cells would include normal cells that are part of your and my tissues that the cancer's recruited to help it grow and fight the immune system. And then even further, in cancers, not every cancer cell is the same. So this is shown behind me here where you see blue, red, white, yellow. These are all cancer cells. But we can tell differences between them. What we don't understand is what those differences mean, and do they explain why some cancers cannot be cured with current therapies. To give you an example about this, and we call this heterogeneity, or another term is complexity, a famous international study of 1,500 people with acute myeloid leukemia, something we consider they've all got the same disease. When looked at for the genes that have gone wrong in those 1,500 acute myeloid leukemias, the researchers came to the breathtaking conclusion that only 2 of the 1,500 looked like each other, and the other 1,498 didn't look like those 2 and didn't look like any of the others. So that gives you an idea of the differences that are seen between individuals across one type of cancer. Now, that sort of complexity isn't there in all cancers, but it is in some. And acute myeloid leukemia is one of the blood cancers with the poorest outcomes for many patients. And so the work we're doing is to try to understand that complexity, try to understand what patterns tell us what new treatments, new targeted treatments to use, to be able to select those treatments for patients, and to be able to understand how well they're likely to work. The way we'd like to conduct this discussion is really to be led by questions that have come in. So I've got a number of questions I'm going to read out that have come in ahead of time through the ACRF. And there's a number of excellent questions coming in the question and answer. We'll try to get to those. And I'm going to lean on Marie-Liesse and Edwin to help answer these questions. But I might kick off with a question that came in well in advance. And that is, how do you choose those cancer patients for selective targeted cancer cell treatments? Well, that's a great question. And we don't actually have long enough for me to give you a great answer. But the short answer would be, we need to see something about the cancer that tells us it's going to be susceptible to a special targeted therapy. And then we need to know how to employ that therapy. We need to know that for a given patient, they're going to be able to tolerate that treatment. And for many new cancer treatments, they're targeted. Before they're available on the PBS here in Australia, they're often available in trials. So trials are a critical part of that. I think the future of cancer medicine will be trying to use more and more targeted therapies to enhance or sometimes replace our existing successful treatments such as chemotherapy and radiation therapy. Another question is, can cancer treatment cure the cancer, or do we still need to operate to remove an organ? In other words, can surgery be enough? Well, for some cancers, surgery is absolutely enough and is pivotal for many of the so-called solid tumor cancers. So the type of cancers that we are researching here are ones that aren't cured by surgery and aren't cured in most people with existing chemotherapy and radiation. So the emphasis in this program is really about the tough cancers where existing treatments don't work. Now, there's a question that's had come in advance. What is this MIBI scope? So, Marie-Liesse, I wonder if you could explain a bit more about the MIBI scope, a critical piece of equipment that's been made possible by this grant. How does it work? Why is it special? What can you do with it that you couldn't do before? And what are you studying with it?
Yes, I'm very happy to answer this question. We've been using that instruments and really getting a huge amount of data that really fascinating. So when we analyze the tumor samples, we look at the cells, and from previous technology, what we could do is identify 2, 3, 4 maybe markers or that enable us to understand what is that cell doing just based on the expression of 4 different markers, 4 different proteins. What the Microscope is enable us to do that is to increase that by 10 times. So we can now look at 40 different markers within a single cell using that technologies. And so by having that intense complexity of understanding of individual cells, we can much better understand their interactions and what the cells are doing. And the beauty of this instrument is not only do we know these 40 markers in that single cells, but we also get the location of that cells. And so we have an idea of all the surrounding environment in which that cells evolve and what are the signals coming from the cells surrounding them and how that will impact individual cells as they talk to each other. And that's really what we can get from this technology. These technologies uses mass spectrometry to quantify the signal that is present in each of these different cells, and uses a beam that actually enables us to locate each individual cells in the tissue. And pictures I've got in the back of me is a representative image of one of the airway of the lung that was scanned using the Microscope, and each individual dot that you can see in different colors are individual cells. Here we're only showing a few markers, but we can make that 40 times more.
Yes. That's a little bit of a good segue to a couple of questions that have come in the Q&A. There's one here, what percentage of patients are impacted by resistance to therapy? I think the best way to answer that is to think about people who, if you're diagnosed with this cancer, at the moment, the chances of being cured is maybe less than 10 or 20%. So unfortunately, lung cancer, the one that Marie-Lies has just talked about, for many people cannot be cured. If it can't be cured by surgery alone. Now, there have been significant advances in new treatment, but the reality is for 80 or 90% of people with advanced lung cancer, resistance is a major problem. It's also a major problem for about 80 to 90% of people with acute myeloid leukemia, for 70 to 80% of people with the other blood cancer, multiple myeloma, for 70 to 80% of people with ovarian cancer, for 90 to 100% of people with pancreatic cancer, 90 to 100% of people with the most common types of brain tumor in adults, and I could go on. And so I hope by answering that question, you get a flavour of why we're focusing on resistance and complexity in the cancers we're studying in this grant. Now, there's a question that had come in advance, and that's a similar question to the one I asked Marie-Lies, but it's about the multi-photon microscope. And so, Edwin, I wonder if you could tell us just a little bit about what a multi-photon microscope is. What can you do with it that can only be done on that piece of equipment that you couldn't do before the grant? And what key scientific question of clinical importance are you trying to solve with it?
Sure, Andrew. So a multi-photon microscope is a special type of microscope that when we shine light onto something, it does it in a way that we can get it really deep into living tissue. Why it's important is we can play a game much like David Attenborough. We can now look and see how cancer cells are growing inside living tissue. We can give drugs that would normally eliminate those cancers and understand that core question of why one or two cells survive that therapy and then regrow and cause cancer. We can watch the cells of the immune system and understand how they're tricked to not be able to detect these cells and eliminate them. And that's a really powerful thing for us to be able to see. You can imagine that we can't see cells around us every day of our life, but certainly to be able to see them deep inside living tissue, understand how they're working to cause these diseases is a huge technological achievement and is shedding light onto a lot of questions. So what we're trying to answer is this idea of why when we give therapies in the clinic, why people relapse, why disease comes back. And it's an extremely complex question. probably different from many different types of cancers. And with the broad range of expertise at WEHI, the idea of having this equipment is to bring those researchers together to answer those questions using this great equipment. So that's the way I'd like people to look at it, Andrew. It's a great microscope that allows us to see things in living tissue and understand these diseases.
10 years ago, we couldn't have got close to it.
We could do it, but we need to look at it akin to the space race, right? We're going further. We're looking at things with better resolution. So what that means when we look at this in cancer is we can look at single individual cells that might go in and cause that disease, or we can look deeper in tissue or really difficult tissue that we've never been able to understand. And as the technology grows and get better, I think, Andrew, we're going to see more tissue and deeper and understand this question a lot better. So that's how I'd like our ACRF members to think of it, is the space race in reverse, where we're trying to look at smaller things rather than bigger things, but we're still trying to understand them with better visualization, better ability to see these events.
Hey, then. Doug, were you coming on to tell me we're just
No, I was going to ask a question, Andrew.
All right.
Is that okay?
It is. There's some ones from the audience I want to get to, but you fire away.
A very quick one, and maybe it links together a couple from the audience. How you're a clinician and you see patients, which is different to Mary-Liesse, Ed, and I. So how do you balance your desire and need to provide new treatments and hope to patients today versus the reality that we as scientists understand, and that is to make these big breakthroughs requires a huge amount of work supported by people like ACRF, sometimes over decades. How do you balance the urgency that patients have now with the kind of patience and resilience and determination you need to work in the lab? Because you do both.
Okay, Doug. Well, thanks for that. Yes, there were questions in the chat about great technology and how long is it going to take before it makes a difference for people. Well, the answer is it will take years, and that might be 2 or 3 years, it might be 10 years. How do we balance this out? Well, if you've got a really tough disease where advances haven't been happening for 10 to 20 to 30 years, then you've got to emphasize research on that, and that's what we're doing. We're looking for major breakthroughs, so not things that are looking to make a 1 or 2% difference. We're looking to make 10, 20, 30% difference. Of course, we'd like to find a cure, but that's going to take multiple steps. The likely outcome of this research is that it'll be applied to how we think about designing clinical trials and applying targeted therapies in clinical trials within the next 3 to 7 years. In terms of what we do for people now who are in desperate need, well, Australia's blessed with great cancer care right across the nation. Even if it isn't going to be the ultimate solution for a person with their cancer, they still will get great care. Clinical trials provide opportunities to try things that are new that might make a big difference. So as a clinician, I regularly offer clinical trials. And one of the things with this research is we're leveraging off clinical trials by gratefully receiving samples from people on clinical trials and looking to see how and why the treatment that they've been receiving on the clinical trials is working, not just in them, but in the laboratory. And that's really important for disease like ovarian cancer. So my colleague, Professor Claire Scott, is looking at this, using this technology to better understand some really tough ovarian cancers. She's not just looking at people where the cancer doesn't respond, she's also looking at people where the cancer appears to vanish and where the new treatment is potentially a cure. So we need to work out not just why treatments may not work, but also why they work unbelievably well in people who may have tough cancers.
Thanks.
There's further questions here that talk about translation. So where do we go from here with the discoveries that will be made with this equipment in the laboratory? Where do we go next? Well, we work closely with our colleagues in the hospitals. So some of this information is going to be important for improving our understanding of the cancer an individual person's got. Marie-Lias, did you want to speak about what we might learn about types of lung cancer and how they might be treated based on research in this program?
Yeah, so we, as Andrew said, we worked very closely with clinicians, and in case of lung cancer, we work with respiratory physicians as well as thoracic surgeon. And so we obtained biopsy from the patients with those clinicians so that we could really then take them and analyze them with those new technology. And what we are really hoping to do is that we can identify by analyzing those samples, whether it is pre-treatment or after treatment, we can identify some changes. And specifically in my lab, we're really interested in immunotherapy. So it is about activating the immune system to tell the immune system, your own immune system, you got to go and kill this tumor. And so we're trying to find ways to activate those cells to go and kill the tumor cells. And so with this technology, we can now say, okay, this tumor cell is near a certain type of immune cells. And this is the type of immune cells that we need to activate because it is sitting nearby the tumor cells. And so by analyzing those samples in details from their molecular perspective, but also where they are located, we can really have a better, find better therapeutic approach because then we know that we are activating the right cells that is near the tumor cells. And so it's really about developing those novel therapies.
In clinical medicine, we call this precision medicine. So, we've got a long way to go, but these are the absolutely necessary first steps to be able to use precision medicine for patients with tough cancers. Edwin, there's a question here from a viewer, Jenny Tetchell. When you talk about living tissue, you say you're using the multi-photon to look at living tissue. Are you talking about organoids, or are you able to look actually into living animals?
That's a great question. So the answer is we can look into any piece of tissue we like. We can take a biopsy from a patient and look at it. That's obviously not living. We can take an organoid, and this is something that I do not do, but there's some great research from many people at the Institute, like Oliver Sieber and others, doing organoids. But I look inside the living creatures. I use preclinical models, which are usually mice that have the mutations that represent mutations that we'd see in humans. And sometimes we also take human tissue and transplant human cancer biopsies and samples, and we'll transplant them into mice so that we can watch them in that context. So yes, Andrew, we look at them in mice. It's quite common to look in other animals like zebrafish, and certainly that's been done at WEHI. And combining that knowledge of human tissue that's been transplanted into animal models and all the organoids, we compile that all together to better understand human disease. I think it's probably important to get an idea of how deep we go into this living tissue. And we go about 0.5 millimeter. And that might not seem too long away, but when we're going through bone and to look into the bone marrow, where of course a lot of blood cancers are, or maybe a spleen where tissue is, or gut for some of the other cancers, it's a long way in an animal. And I think what you'll find is, Andrew asked me before, what's going to happen with the technology that we have now and moving forward to technology in the future. I think that we'll get deeper and deeper into that tissue and get better answers about maybe structures that we haven't seen before, or maybe where those cells are moving throughout the body.
Thanks, Edwin. And of course, it's important to note that this is painless for the animals. They receive an anesthetic, so it's painless. There's a great question in the chat here. How long has cancer been causing disease in humans? Well, we think since time immemorial. There's absolutely records of cancers and big tumours back in Egyptian times, Greek time, ancient Chinese and Indian times. So it's just part of the human condition. Cancer is becoming more common. And the longer we live, the more likely we are to have cancer. So I don't think there'll be a time where the human race is cancer free. But increasingly we're seeing many healthy people who've been cured of cancer. And so it's something in their past. We still have many people with cancer as part of their present. And our hope is that for some of those people, the work we do here will have an impact. I think most likely the biggest impact will be people who are yet to be diagnosed with cancer. And potentially for future generations. There's another question. Does this research only involve Australian scientists' research teams? Well, it's centred here in Melbourne, but it reaches outwards. We have international collaborators. Murray, yes, did you want to talk a little about international collaborations that you've been involved in already in this regard?
Yes, absolutely. Yeah, definitely we have international collaborations with mostly the UK and also in the States in my case. And I guess the advantage of this collaboration is really taking advantage of accessing more patient samples and biopsies that can be done in slight different ways depending on the different clinical care in those different countries. And that really enables us to access a more diversity of samples or samples on clinical trials that are done somewhere that is not necessarily done here. And so we do have those collaborations to really being able to access more tissue samples and different category of samples, I guess, yeah.
And pancreatic cancer is one area where we're involved with local, national and international collaborations. And increasingly, we understand the genes that go wrong to lead to pancreatic cancer. So we're working on understanding how the cancer cells relate to other cells in the tumour and to give us some clues. So as yet, no breakthroughs for therapy, but much better understanding of the problem. And hopefully, we're doing experiments that will show how to tackle it in the future. There's a question from Natasha Lee. I've answered part of that of her question, but she's asked, are multiple treatments needed for the different mutations in a cancer? Are these treatments given together or separately to monitor the response? Yes. Most cancers will end up needing multiple treatments, often given together to get the best results. And with targeted therapies, I would say we're at the dawn of era of understanding how to use these new smart anti-cancer drugs, and that increasingly over the next decade or more, we'll work out how to use them in combination so that having 2 or 3 mutations isn't a barrier to cure. And that, in part, is the essence of this program. That brings us to the end of the discussion. I want to thank Edwin and Marie-Liesse, but most importantly, I want to thank the audience for the questions that came in before we started and during it. Back to you, Doug.
Thanks so much, Andrew, and thank you to the audience for your wonderful engagement and really thoughtful questions. Thank you also to Ed and Marie-Liesse for your contributions. Kerry and Russell, can you turn your cameras on? It would have been really lovely if we were able to do this face-to-face, not just with you and your staff, but also with all of the supporters that were in Melbourne. And great that we can have supporters of the ACRF from interstate joining as part of this. If there's one kind of silver lining to COVID, it's that we've got a lot better at being inclusive in the way we run events, and having people from all around the country and overseas join this event has been really special. Russell and I were lucky enough to be able to meet last week, and we did the usual routine of presenting the large check. And I was able to show Russell around the facility, as you saw earlier. And I'd like to play a video that just captures that important moment of ACRF placing such trust in us. Well, that was short and sweet, as it was on the day, Russell. So thank you so much for coming down and visiting, and thank you for being the ACRF's ambassador as we, formally opened the facility. I'd like just to finish off now by thanking the ACRF and all of its supporters and donors. I hope what you've come to appreciate is that making advances in cancer takes really special people to come together. If we're going to beat cancers, it requires scientists who really want to work together and work together across disciplines between basic science and clinical care, where the science informs the clinical trials and the clinical trials in turn form the science. But I guess what we've realized is that even when you have these exceptionally talented people like Andrew and Marie-Liesse and Edwin, who have thrown their lot in together, who've decided to collaborate, not just for years, but decades, that's not enough. In order to make the real differences, we need the support of the community. And to me, ACRF and its supporters are really the best example in Australia of organisations driven by passionate grassroots patients and families who have really borne the brunt of cancer supporting the scientists. I think that is a wonderful virtuous cycle. And I would just urge all of the supporters to stay in contact with the scientists that you're supporting, stay in contact with the ACRF, because I absolutely know that together we can make a difference and drive the improvements in terms of patient survival and patient well-being, and therefore reduce the burden on patients and their families. So thank you to the ACRF, thank you to my wonderful team here at WEHI, and most of all, thank you to the passionate and generous supporters and donors of the ACRF.
Thank you so much, Doug. Thank you, Andrew, Marie-Liesse, and Edwin. I have to say, I'm absolutely in awe of your exceptional talent, and thank you in advance for the impact that you're actually going to have with this equipment. I'm absolutely sure that future generations will be in a much better place thanks to you. At ACRF, we talk about backing brilliant, and I think today's opening has demonstrated all the elements that we look for. Brilliant people with brilliant ideas, brilliant science, brilliant technology. As you say, Doug, it would have been lovely to be there in person. So I'm so thankful to Jane and Casey from WEHI, to Carly, Isabel, Cheryl from ACRF, as well as our amazing fundraising and support teams. They really do a fantastic job. And as you say, Doug, the downside of not doing it in person, the upside is that we can broadcast nationally. I'd like to quickly acknowledge we had a couple of other board trustees online today. So Greg Cam, Peter Jones, and Sir Angus Houston, thanks for joining. Again, to OCRF, we so value our ongoing collaboration for all funding projects that involve ovarian cancer. This really drives some efficiency into the funding of cancer research, which is very important to us. But most importantly, a huge shout out to ACRF supporters. You make this all possible, and I cannot thank you enough for enabling this brilliant cancer research. Our next opening will be the Children's Cancer Liquid Biopsy Program, which was awarded at the same time that this grant was awarded. So that'll be on the 11th of May, and I look forward to having you join us then. Cheers. Thanks, Doug.
Thanks, Kerry, and goodbye, everybody. Thank you for joining us.